Dear everyone, it's my pleasure to welcome you here this evening to this event about transient global amnesia. And I would like to introduce the speakers tonight before I talk a little bit about this very fascinating syndrome. So tonight you will hear from myself, since I'm the first one on the top. I will also give an introductory lecture. My name is Tan Doan. I'm a neurologist at St. Olav Hospital and a researcher at NTNU. And then Tobias Navarro-Schröder will continue. He's an associate professor at the Kavli Institute for Systems Neuroscience, followed by Gøril Rolfseng-Grønnevet, who is actually my boss at St. Olav Hospital, the head of the neurological department, and also an associate professor at NTNU. Gøril is doing research about memory as well, but in the context of Alzheimer's disease. Then Alexandra Galvas-Töcan will continue. She's doing her PhD with Tobias and I at NTNU. And Livia Ole Hagermark is currently doing her research medical line at NTNU as well with Tobias and I. And we are also delighted tonight because two persons who experienced TGA as late as last year were keen to come and also give us another perspective on this very interesting syndrome. So we'll have Arild Bleke-Saune and Margaret Ovid. Here is the program for tonight. It's not written in stone, but we'll try to keep up with the time. And of course, we can take questions also at the end of each presentations, but there will be a dedicated question and answer session at the end too. And both people here on site and people online, I believe, may be also able to ask questions, since this event is streamed. So let me start with transient global amnesia. And I will start by asking you a very simple question, which is, how does it feel to lose the ability to remember within the flash of a light bulb? It's a very strange question, but actually, in Norway, about 300 individuals experience this every single year. And given this incidence, it means that actually worldwide, someone experiences this phenomenon, acute memory loss, every 72 seconds. And what's even more striking, I think, as a clinician at least, when these patients come, is that no one knows why it happens. But hopefully it's not a chronic condition, it's a neurological syndrome, it's a conundrum. We still don't know why it happens, but it lasts a few hours. And that's essentially what constitutes this transient global amnesia. I will talk a little bit more because it probably sounds a bit abstract to you. But before we jump in, I have a very simple question, which is also, how do we remember? How does the brain encode memories? Well, it does not happen everywhere in the brain. There are some circuits that are dedicated for memories. We collect information about the surroundings, and these memories are also influenced by our state of mind and emotions. But memory can be essentially split into different components. We often split it into implicit or non-declarative memory, which is, for instance, when you first learn to ride a bicycle, it took a little bit of time. It's procedural memory. And it can be also split into explicit or declarative memory, which is probably what you think about when you think about memory. I will make it a little bit more complicated, but I guarantee it will serve the purpose of the rest of the lecture too. So we have, within explicit memory, something that is called semantic memory, which is a general knowledge of the things that are here in the world. The fact that we call this a table here, a chair, a screen, and be able to use these different words and concepts around us in order to navigate efficiently. And then we have our hot topic for tonight, episodic memory. And what you see here, which is essentially a small extract of a beautiful documentary about the city here, Trondheim, Trondheimsreisen, is this episodic memory, which are memories that have a certain content, for instance, doing ski jump, as you can see here, or playing football, and that were found in a specific place, for instance, in the mountains or a stadium, and that occurred at a particular time point. So you can see that these memories have a what component, a where component, and a when component. And that's the very important type of memory we will talk about tonight. So now we jump back to our topic of interest, TGA. I picked up a case study because I think it's easier to understand a little bit if we have something to relate to. And we could take, for instance, a person around his or 60s, 70s, doing some gardening, and everything is fine, and suddenly some things happen. This person experiences maybe a little bit of dizziness and feel weird and cannot remember exactly what he was doing and why he was doing this and what time is it now. He starts to wonder, which day is it? Why I'm here? What am I doing? Why do I have this thing in my hand? And that's essentially what people who experience transient global amnesia or TGA feel. It's very puzzling. And it's very frightening. So there is a collection of symptoms that are very stereotypical for this syndrome. And it affects memory, but not all the type of memories. This specific type of memory I mentioned called episodic memory. And you have something called a total anterograde amnesia, which means that there is no ability to form new memories, and a partial retrograde amnesia, which is essentially a difficulty to retrieve older memories. And because of this, it's very hard to remember things. There is a delayed recall and a tendency to ask the same questions in loop again and again. Otherwise, the consciousness is preserved and there are no specific neurological deficits. What does that mean? No paralysis in the arm, there is no numbness in general. It's, the body is working, everything seems fine, but the memory is specifically impaired. And self-identity is preserved. That's also a feature. You remember who you are. Now, it lasts a few hours, and everything seems to revert back to normal within 24 hours. It's actually a diagnostic criteria. There are usually identifiable triggers. In the case of our patient here, or subject, it was doing gardening, so physical exertion. But it's not only with gardening. It can be anything from jumping into cold water to have sexual intercourse, which is actually very normal as a trigger for TGA. It usually affects people in their 60s, and there must be something with the brain happening at this time which make certain people very vulnerable for TGA. It affects 3 to 8 people per 100,000 inhabitants worldwide, and it has been told that it's a single occurring event, but actually, new research has demonstrated very convincingly that 1 out of 8 people will experience TGA again. And moving on to the after effects, after 24 hours, we usually think that everything is back to normal. But if you test well certain cognitive domain, certain aspects of memory, some subtle deficits actually persist from days to months. And the research is not very consistent here. The spectrum, magnitude, and duration vary between studies. But until now, we think it's fully reversible. And of course, into a very busy clinical life, as we are, have to deal with when we are on call, we think it's a benign condition. The prognosis is excellent, and it's nearly impossible to demonstrate that there are long-term deficits. So we think that the syndrome is benign. You will hear a little bit more about this from Livia later, because that's something we would like to investigate. Now, TGA, it was not first described by Théodule Ribot in France in 1881, but he was one of the first persons to explain how amnesia affects more recent memories than older memories. And actually, this concept is very predominant in the field of memory research, and it has even kept his name as Reebo's law. When you lose memory, the recent memories are affected because they have not been consolidated yet. One of the first person actually to describe this, not very consistently, was Robert Benon, a French psychiatrist who introduced the term ictus amnésique to describe transient episodes of memory loss in patients. But it was just a few case reports, not very systematic description. So we had to wait until 1956 that another French psychiatrist, I'm sorry it's all about France here, but you will see soon, Jean Guyotat, described a large case area of patients. Fine. France is not the center of the world, actually, so it has been happening probably everywhere. And exactly the same year, independently, Maurice Bender described also a large series of cases that seemed to indicate patients who had TGA. But the real pioneers who actually coined the term TGA for Transient Global Amnesia were two eminent neurologists, Miller Fisher and Adams in 1964. They described a lot of patients. And actually, if you think a little bit about this, oops, I'm sorry. If you think a little bit about this, maybe you are wondering why this Robert Benon is looking so weird. And actually, it's not Robert Benon. I spent hours trying to find a picture of him. It's because it's impossible. And because most of the people in France published in French. And if you want to stay in history, and publish and be read and be recognized, you have to publish in English. So TGA, do we get any trace in our brain? Maybe. When you come in the acute, in the emergency room, you can get small traces. So if you slide through the brain here and you zoom in, you can see these little white dots into something called the hippocampus. Sometimes, with standard MRI, magnetic resonance imaging, which is an imaging modality we use as clinicians, we can find these lesions in hippocampus, and they're fully reversible. But recently, in 2020, we got this amazing machine here, this very powerful MRI, 7 Tesla, state-of-the-art, unique of its kind in Norway, and it's here, right around the corner. So we were very happy because we could demonstrate using this machine, actually, that we found a lot more lesions in people who presented with symptoms similar to TGA. And it was a nice way to diagnose, because these lesions are pathognomic. If you get these lesions, you're pretty sure you got TGA. So I talked about something weird, hippocampus. Maybe I should stop a second here. So I have a brain here. I will just make a cartoon of it and dig a little bit in the temporal lobe here to make appear this blue structure here. I'll just zoom in and show you why this blue structure is called hippocampus. It looks very much like a seahorse. And this seahorse is not proper to humans. We tend to think we are uniquely cognitively competent, apex predator, all these things, top of civilization and so. But actually our brain are not so different from the brain of other animals, from non-human primates or even rodents. So if you slice through our brain and you slice also through the brain of a monkey and a rodent in this structure, you can see that the fine architecture is very similar. Maybe it sounds heavy, but I will just go through the substructures here. We have something called the dentate gyrus, highlighted in yellow because it looks like a tooth. Something called the cornea ammonis, CA1, CA2, CA3, different subfields that are very important in the hippocampus. And something called the subiculum for support. And all these regions together form the hippocampus, or hippocampus proper. And then there is a little neighboring region, a little area called the entorhinal cortex that you will hear more about tonight. Now we move on to this hippocampus and how it's organized. So I'll just take it out like this and magnify, and then pull it flat out. First, just isolate it and pull it flat out, because it's easier to understand. And what you can see is a bunch of subregions that are connected to each other into a very specific way. It's not a weird wiring patterns, it's very unidirectional. And actually, when you get sensory inputs from the world surrounding you, your entorhinal cortex, or EC, is sampling, changing the meaning of it, and feeding it to the hippocampus. And actually here in Trondheim, there has been very important discoveries. First, in the UK in the 70s, finding very important cell types in CA1 in hippocampus, and here in Trondheim in 2004, 2005, with the discovery of entorhinal grid cells by Professor May-Britt and Edvard Moser. And collectively, these discoveries were awarded the Nobel Prize, something we are, of course, very proud of. Now, this area, CA1, is of particular interest because every lesions in TGA actually appear in CA1. And CA1 is like a crossroad in the hippocampus where all the information funnel and gather. And why it's relevant? Because it's very, very demanding in terms of energy. It has to process a lot of information. And probably the blood flow is very important. So one of the hypotheses that TGA occurs is that the vessels, the arteries feeding oxygenated blood to the hippocampus, and the vein draining the oxygenated blood, there is some kind of congestion, or maybe there is some kind of obstruction with the blood flow, and then this little area that really needs energy does not get enough energy, and you get a mini stroke, and you get these tiny lesions of TGA. That's one hypothesis. The other one is that we have a chemical in the brain called glutamate, which is the most common neurotransmitter or chemical in the brain for brain cells to communicate with each other. And it seems that there is, in these lesions, a lot of release of this chemical. And because of this very massive release, we get something called glutamate excitotoxicity. So why TGA is so fascinating, and why are we thankful to have the opportunity to talk to you here, but also to you online about this, is that for me personally, at least as a researcher and clinician, TGA is a neurological conundrum. It's so old, yet it's so little known, and hopefully, here together, and also with some tests you will hear more about, we could potentially push the frontiers of knowledge and perhaps understand TGA a little bit further. TGA did not start in 1881 with Ribot. Probably caveman experienced TGA. The brain has not changed so much. And despite 150 years of research, we have still no clue why it happens. And more importantly for tonight, perhaps, is that TGA is a window to what makes us human. It strips away the scaffolding of our memories for fleeting moments, revealing the fragile architecture of identity. And in that brief void where time slips and personal history vanishes, we catch a glimpse of how deeply memory defines what makes us human. On these words, I would like to thank you for your attention and invite my good colleague Tobias to join the scene.
Test 1, 2. Yes, it works. Great. Thank you, Tan, for that beautiful introduction. So, in science, it can be really beneficial to combine different perspectives, to use different methods and work together across disciplines, and also to study different animals, different species. In my work, I like to translate findings from studies done in rodents, in mice and rats in particular, to humans. And I've actually started doing that already when I was about 17 years old in a high school project, where I was lucky to have a biology teacher who had connections with the university, and she enabled us to have this developmental study with mice that we reared in different conditions. With 3 conditions: deprived condition, enriched, and super-enriched. And in the deprived condition, the mice were living in really just a small box with wood chips, and they had food and water as much as they wanted. In the enriched condition, they had a little house in addition, where they could hide under and climb on top. And in the super-enriched condition, we went wild and built them basically an adventure park with multiple levels and branches and tunnels and buildings, and a mesh wire where they could climb up. So it was really quite a big environment. And after a few weeks, these animals grew up to become adults. And then we did various cognitive and memory tests. And what has been known since a long time is that these enriched environments can really help cognition and memory. And that was not even the most striking thing that I noticed. What really sticks in my memory is the differences in anxiety, actually. So the animals in the deprived condition, when we put them on what is called an elevated plus maze, so that's a maze that is elevated from the ground and has basically, it looks like a plus from the top, and it has 2 open arms and 2 closed arms. And the mice like to hide, so they will usually go into the closed arm, and after a while, when they calm down, they become curious, and they usually will explore the open arm. The mice from the deprived condition were curious enough to explore the open arm, but in many cases they were basically terrified, or they looked terrified to me. With their legs extended, they were just going very slowly, looking over the ground. They've never seen an abyss like that. And then in the super enriched, in the enriched condition, mice were a bit quicker to explore the open arm, so they seemed less anxious. And strikingly, in the super enriched condition, there were really some super mice coming out that were not only, they seemed not afraid at all, but some of them even managed to climb down the whole construction. They basically, this experiment wasn't designed for mice like that. And they managed to, some of them managed to escape, which is probably the reason why this would never happen again in the school, this type of experiment. But it really showed that an enriched environment can lead to improved cognition and memory and drive exploration. Now, why do I say this? To me, Norway is kind of an enriched environment. It's blessed by nature, a lot of wilderness, and the whole world looks to Norway for examples on how to do education and how to organize the economy and the society. And also for science, as Tan mentioned, there are great opportunities, also particularly here in Trondheim, with the 7 Tesla MRI machine, which we got a few years ago. And what is magnetic resonance imaging? So it's basically making images of the tissue without having to cut it open. We can look inside the brain using a big magnet and radio waves, and you can use it to measure different things, for example, the structure or also the function of the brain, how active a brain region is. And with the strong magnet, the 7 Tesla magnet, we can really measure with higher and higher resolution, which is very beneficial, as Tan mentioned, for example, for TGA to detect these tiny lesions in the hippocampus. Now, Trondheim in particular is a super enriched environment to me because, on the one hand, the Kavli Institute that Edvard and May-Britt Moser really built and are keeping to build and create new research groups with different focus. So we have people that study zebrafish, mice, rats, and we do mainly humans. And then we try to really work together to answer the questions about memory that are fundamental and basic. So our favorite brain region perhaps is the hippocampal formation that Tan so beautifully described. And it's very useful for translational research because the basic organization is quite similar across different animals, different species. So we can learn a lot from looking at rats about the human brain as well. Also people like Menno Witter, leading neuroanatomist of the hippocampal formation, was really influential in my work. Why do we need more basic research on memory disorders like TGA and Alzheimer's disease? Well, society is aging, and by 2050, it is projected that the prevalence of dementia will double, and that comes with a burden on next of kin and society as a whole, the medical system. However, we don't really understand very well how memory actually works, or how the cognition and brain really work in the brain. And then we need basic research to understand these fundamental mechanisms without promising that we can solve a certain disease and cure a certain disease tomorrow. Things need time. One example for the benefit of basic research, also for potential clinical applications, is the following. So strikingly, Alzheimer's disease pathology actually starts in this region called the entorhinal area, which happens to be the region where also the grid cells are found that are so famous, and that Edvard and May-Britt Moser were awarded the Nobel Prize together with John O'Keefe for the discovery of Pay cells and grid cells in 2014. So that happens to be exactly the region where also Alzheimer's starts in the beginning. And together with my then PhD advisor, Christian Döller, we ran a study using a method that looks at so-called grid cell-like representations in fMRI, which is a putative fMRI measure, functional MRI measure, for the activity of these grid cells. And we found that people with genetic risk for Alzheimer's disease had weaker grid-like representations, which then sparked the idea that maybe there could be something like a biomarker to diagnose Alzheimer's earlier, which would really be important to develop a treatment. Follow-up research has then, or is still being done by Güril Grönvet and Surabonovi, Marcia Beku. Güril will talk more about this in the next talk. So let me try to be quick now. When Tan approached me with the question, should we study TGA, I got very excited and said, yes, let's do that. We applied for funding, very grateful for Helse Midt Norge, Samarbeidsorganet, who funded our project. Also people like Guillermo Genovese and Axel Niemann were instrumental in setting it up. And you will hear more by Alexandra Galva-Stöckhan and Livia Ole Hergemark later about the precise study. We acquire various types of data. I won't go into detail now. Various types of cognitive tests. The take-home message is that the link between our behavior and memories with the activity and metabolism of cells in our brain is not well understood. Importantly, we don't know what we don't know. And if we want to explore an unknown space, we have to have basic research. Free researchers that, free of stress, can use advanced technology to answer deep questions. And basically, we need super enriched environments for excellent science. I'm grateful for Edvard and May-Britt for building up the Cavell Institute. And if you want to participate and support our research, there's the research fund of the Cavell Institute where you could support us through a VIPS donation, for example. And we will do this prospective study on TGA, which Livia will talk more about. So this QR code will keep popping up if you want to scan it later. This is for people that had TGA at one point in their life. Thank you very much for your attention.
So, maybe we can go to the next presentation. Oh, I can just click. Perfect. That's great. There you go. So thank you very much for a nice invitation to come here and talk today about my favorite topic, which is not TGA, but that's also very interesting. But my main topic of interest is Alzheimer's disease. I have 8 minutes, so it's going to be at a glance. So I guess most of you have heard about Alzheimer's disease. It's the most common cause of cognitive decline and dementia, accounting for approximately 70% of all cases. And that means that approximately 70,000 people in Norway are living with that disease. And I'm going to repeat some of what Tobias already mentioned, but repetition is good for memory, so maybe you will remember it more. So if you look at the figure behind me, so we don't know why you get Alzheimer's disease yet, and we still don't have a cure, but we do know that the number of individuals living with the disease is increasing, like Tobias said. So you see the figure here that in 2005 and compare it with 2050, there is an incredible increase in the number. And one of the reasons is that we are getting older, but there are probably several other factors that are still unknown to us that we have to explore. And if you look closer into a brain of a patient with Alzheimer's disease, you will see that they have an accumulation of 2 different types of proteins. And those 2 proteins are called amyloid and tau. And these 2 proteins are present in every normal human brain. It's present in every brain in this audience. But for some reason that we still don't understand, during Alzheimer's disease, these 2 proteins will accumulate and form plaques. So you will have amyloid plaques and what we call neurofibrillary tangles. And also, if you look very closely to the brain, the specific region where this pathology typically starts is exactly in the entorhinal cortex that Tan mentioned before. And this is the region where we know that the grid cells are located, the very famous grid cells, or Nobel Prize-winning grid cells. But later, the Moser group also discovered that time cells are located here. So cells responsible for the perception of time. And knowing that Alzheimer's disease starts in this region, if you ask a patient with Alzheimer's disease what type of symptoms they experience, they will tell you that they have problems in remembering what's happened, so short-term memory. They will have problems in remembering where it happened, so spatial navigation. And they will have problems in remembering when it happened, so time orientation. So this goes back again to memory, spatial navigation, and time orientation, which is actually basically the link also to TGA. So the symptoms are quite similar, but in TGA cases, they resolve within a few hours. But for Alzheimer's patients, these symptoms will gradually increase. Alzheimer's disease is a continuum, so what does that mean? Well, it means that it takes several years with accumulation of these two proteins in the brain before the patients experience any type of symptoms. 15 to 20 years to be exact. So that's the time where these proteins start to accumulate in the entorhinal cortex and the other regions of the temporal lobe. And after 50 to 20 years, the patient starts experiencing symptoms. And in the beginning, the symptoms are subtle, so they have a somewhat cognitive decline, but they are perfectly capable of independent living. But after a while, the symptoms will continue to develop and they reach the dementia phase, where the symptoms have evolved to such an extent that they need help in activities of daily living. This is the slide that Tobias showed as well. So in Trondheim, we have the unique situation that we have the Kavli Institute with world-leading expertise on the entorhinal cortex, so the normal function of this brain area. We have the Neuroclinic where I work and Tan work, where we see and diagnose patients with Alzheimer's disease and do clinical research on patients with Alzheimer's disease. Then you have the 7T and advanced imaging techniques. And that gave us the idea that we could collaborate on research also on Alzheimer's disease. And it's also very practical because we're just a few meters apart. So that facilitates the collaboration even more. And we do something that's called translational research. And what does that mean? Well, it means that we study all the way or explore all the way from the cell level. So we look at individual cells and brain cells and neurons, and we can actually develop specific brain cells from patients with Alzheimer's disease. And we collect that knowledge and use it into animal models, both with Alzheimer's disease and healthy animals. And we collect that knowledge up to the humans that we do research on, so Alzheimer's disease patients and healthy controls. And we use that knowledge back down again via the animal models and down to the cell level. So translational research means that you try to use the basic neuroscience and then convert it into the clinic and vice versa. And that's basically essential in trying to understanding the disease more. This is a very busy slide. I'm not going to go into detail, I have 8 minutes. But this is just to show you a small glance on how diverse we need to be in order to try to understand the disease better. So we go all the way from looking at different metabolic patterns in the brain, so what happens in the brain, metabolism during the disease. We look at genes, genetics, expression of genes, and how the genes are regulated in patients with Alzheimer's disease. We look at possible protective factors that can protect us from the disease and how we can make or increase the levels of these protective factors. We'll look at cell levels. We'll look more specifically into grid cells and time cells and their location in the entorhinal cortex. And we've also developed a method to look at brain organoids, meaning that we take blood samples from patients and we're able with very specific methods to make small mini brains or brain organoids from the blood cells of the patient, which gives them a very, very interesting small brain model of every single patient that we do that on, that we can do experience on. Because when you look at humans, we cannot open their brains. We need to find other models to explore and do research on brain level. And we had, for some years ago, we had an evaluation from our scientific advisory board, which was quite positive, and we took that with us and are going to work further. So it was nice to hear that others are believing in what we do. And it's not only me involved in this, so it takes a village. So all these people here, and probably more of them also that I haven't been able to put in, are involved in this research center that we have, and we're also going to continue with the work in a new research center from October this year. And I think that's my time, so thank you very much.
Thank you very much for a nice introductory lectures of my colleagues. My name is Aleksandra Gałwa-Stekan, and I'm a researcher at Kavli Institute and a medical doctor at St. Olaf's Hospital. And we're coming back to Transient Global Amnesia, because if you're gonna come to the St. Olaf's Hospital with Transient Global Amnesia, there is a big chance you're gonna meet me. I'm not alone. I work with two other colleagues, also with Tan and Axel, and we're actually on the call all the time. We are in a very good cooperation with emergency medicine doctors who tell us when somebody with TGA is coming, and then we are arriving. If the patient agrees, we include them in our research, but we perform medical examination like on any other patient we would normally. We don't treat patient worse just because they don't want to be a part of a research. Our research is longitudinal. It means that we observe one subject or one patient through few time points. In our case, it's we observe a patient immediately. We also observe them 24 hours later, and if they are kind enough, they're coming 14 days later to get the control. And what do we do with those brave patients who want to be a part of our research? As mentioned before, we have a super machine, 7 Tesla one, which allows us to get a much better quality of the pictures. And there is not only structure we can see in MRI. We can also look into how the water is moving inside the brain. What's the function? We can look at the chemical and mechanical properties of the brain, which helps us to understand the TGA better. In addition to all those, we also perform cognitive testing. And what is cognition exactly, could you ask? And it's basically an action in which we acquire knowledge through experience. And the tests we are taking are, among other, the ones in which we are testing memory, the ones in which we're testing verbal fluency, decision-making, and also checking on the patient mood. And it sounds complicated because it kind of is a logistic, not nightmare. I don't want to say nightmare, but it's quite complicated logistically. I'm gonna add to that map we were using before one more place, because that's where the patients are arriving. And that's where we get help from our colleagues from acute medicine to let us know and first see a doctor. That's where the patient first meet a clinician. Then we are coming into the picture. Here is our super machine, so we need to schedule scanning and talk to radiographers and radiologists very often. We cannot just put the patients on the corridor, so that's why neurological department is coming into the picture, which is also extremely helpful, and we are very grateful. And when we collect the data, we can analyze it there in the Kavli Institute. We included so far 49 patients. Maybe it doesn't sound like too impressive, but actually considering that there is 300 patients per year in Norway, I would say it's quite impressive. We started in September 2022, And expected incidence, which was named already by Tan in his lecture, supposed to be 3 to 8 per 100 citizens. And so far from our study, it seems that it's almost as double in the, considering like the higher number already. So, okay, we have all those patients. Like, well, what do researchers do? Well, we ask questions. So first and foremost, we are wondering, how does the TGA happen? And my particular question I'm working on is there a chemical imbalance in the brain during TGA or before, which is a cause of it? And why some people do get TGA and some are not? What's special about them? Do they have maybe that imbalance from before? And the last question is like, how does it affect brain function? Is there any consequence for the patient experiencing TGA? What's for the person having it in it? And like one of the, as Tam mentioned, one of the hypotheses which we are wondering about is the glutamate one. And you may ask, what is glutamate? If somebody from here, or like most of people from here, I assume, are from Norway, you probably had gastromat in your life. Because glutamate is amino acid, which or MSG, if you ever heard about it, maybe you were scared that you're supposed to avoid it. But that's amino acid, which is also a neurotransmitter in our brain. We can also find it naturally in mushrooms and in tomatoes. It brings a delicious flavor to our food. And it's actually the most abundant neurotransmitter in our brain, which stimulates another neurons to get excited. And as with everything in our body, it needs to be strictly controlled when it comes to where it is and how much of it is. Let's talk a little bit about what we have when it's too much of glutamate, because like with everything in life, when something is too abundant, it's not too good for us either. So when we release too much glutamate into the synapses, this is my beautiful diagram in which you can see the presynaptic neuron, brain cell, and postsynaptic, so they like connect into that little bridge. Here around you have an astrocyte, which is kind of hugging those neurons and taking care that they have it well and that they're comfortable. So what's supposed to happen, it's like we release that little information, that little email one neuron is sending to the other one, to that little door the other neuron is having. It's like, it's time to get excited, it's time to get stimulated. So while this is happening, astrocyte's supposed to take it back. When it's too much of it, we let too much gas into our door, and the cell might be swelling, and you can see it as the thing which we probably see in here. It's like too much water in the cell. So we have a technique which allows us to measure the amount of that neurotransmitter, and we found out that it's much more of it at the beginning, and it's dropping slowly, while when patients are coming for the control, it's much lower. And our brain regions are not in vacuum; they all are connected vessels. So my other colleagues, Suniva and Helena, are working on how this part of the brain is connected to the other parts of the brain, how it's like those little highways are connected. And they actually found out that there are many connections between that part of the brain and the rest of brain, the other regions of brain, which have many other different functions. So what I want for you to remember, that the take-home message is like increased glutamate levels might support the hypothesis of the glutamate-induced cytotoxicity, which could be the key element to explain why TGA is happening. Stay tuned for more results because we are collecting more data, we are taking more tests, among other more cognitive tests and like other techniques like elastography. And it's ongoing research. The data for now is preliminary. We still need to analyze it more and work on it more. But I think it's something to get excited about. So thank you, everybody who went through my hands. Thank you for helping us to make science. And if you still want to make science and you had TGA, that's the QR code for Livia's research. Thank you very much.
Thank you, Alexandra, so much for your great presentation. Now, moving on, I will talk about the new study that we will conduct on previously patients who have previously experienced TGA. My name, as Tan mentioned at the beginning, is Livia, and I am a medical student currently attending the research program in Trondheim. So for this study, our aim is to have one of the largest, if not the largest, TGA study done to date. And to make this possible, we want to conduct this online global study to include international patients, because as Alexandra also mentioned, we only have 300, about 300 each year in Norway. So to take it online and globally, we will hopefully receive even more participants. And this study is also a collaboration with scientists from the University of Oxford with Professor Masud Hussain and Dr. Sija Sao, who've helped us develop the test we will be using for this study. So why do we want to study TGA afterwards? Well, as Tan mentioned, it's transient, and well, some people, or many people, think of it as totally benign, or that it goes back to normal after the episode. But actually, seen in some studies and research, that there has actually been some lasting effects on both verbal memory, but also spatial navigation abilities. So therefore, we want to, with this study patients who have, or people who have experienced TGA way back, even years after months, weeks, and look if there is any lasting effects, and also see what cognitive domains are affected. Like we know about episodic memory in the acute phase, spatial navigation abilities, recalling abilities. And then we also want to look at how the brain recovers, because in TGA you have impaired memory, and then the memory or the ability to form new memories will come back. And why exactly do we want to study TGA? Why does it matter? Well, TGA is quite unique, so it gives us a window to study both impaired memory and also recovered memory, and also the recovery process of memory. And this study will mainly focus on the after effects of TGA, where we will compare people who have experienced TGA and how they perform on cognitive tasks to healthy controls. And also, as Tobias mentioned earlier, the world is aging. We have, with the better medical progress, and also just publicly better health, we have an aging, or people are getting older, and therefore memory impairments such as Alzheimer are becoming more and more common. So that leads us also to the goal of this study. Why would we want to know more about memory? Well, of course, since the memory impairments are becoming more and more common, it would be highly valuable to look at the diagnostic, get better diagnostic, better care for general memory impairments. So for this study, what will the participants do exactly? So you would have to use a smartphone or a tablet to complete these studies, as the tests are designed to be completed on a smartphone or a tablet. They're not designed to be used on a computer. And they're also dependent on internet connection, so ensure stable internet connection and also have a quiet space while completing the test. And as mentioned, the testing platform is developed by researchers from the University of Oxford. And I will just quickly go through how you can start tests, since there are probably a lot of people who want to partake in the study. So how you would start by clicking on the QR code or the link, and then you will get right into the page, open it in like a good browser, and then you will select your preferred language. So far, we only have it in Norwegian and English. And then your participant's ID should already be pre-filled when you start. And then you will enter your age and your gender, and then click Submit to begin the testing. And then there's a pretty clear walkthrough of everything you will do when you get into the test, but I'll also show you just briefly how the test will look. So the first thing you'll see, it will be a white circle with a dotted black line, and then you will tap it as soon as it turns black. As you can see here, it's already turned black to test the reaction. Then you will be given a set of numbers, which you have to remember right afterwards, but also later in the test. And the same principle goes for the figure, where we will see 3 blocks that you have to combine to recreate this figure, and you will also be asked to recreate it later. And then you will see a background with a misfit object that you will later have to do the correct object in the correct placement for the correct background. And then you will see 8 symbols with 8 corresponding numbers, and when a symbol pops up, you have to find the correlated number and do this as fast as possible. So this QR code has already been showed multiple times in this presentation, but yeah, you can scan it to get right into the test and start right away, and it would also pop up later if you want to scan it at a later time. And after you're done with this test, there will be a questionnaire that will contain questions about your TGA episode, asking when did it happen, were there anyone with you when it happened, has it happened multiple times? And we will also ask for some health information about you and general information like, are you left-handed, right-handed, what's your height, and also ask some questions about anxiety and autism. These questions will not be used to diagnose you in any way, just to see if there's any similarities or correlations between it or with TGA. And after you're done with the questionnaire and everything, if you would like to even participate further, you could play a game called Sea Hero Quest. This is developed by the researchers at Alzheimer's Research UK. Here you will download, if you want to partake in this first study as well, you will have to download the Sea Hero Quest research app, either on App Store or on Google Play. And here we will ask you to complete at least the first 15 levels, and you will have to enter a personal key when you start, but this will also be shown after the questionnaire and this mini-octal portal. For this Hero Quest, you will be tested, your spatial navigation abilities will be tested in more real-life scenarios where you will be driving a boat around in different environments. And first, you will test your spatial navigation abilities with wayfinding, where you will be shown a map with 3 different landmarks that you have to remember. And then you will guide through different environments and find all of these landmarks. And yeah, you will give a star 1 to 3 on how well or how fast you completed the task. And you can do the levels multiple times, but it will show, like the data will show if you did it multiple times or just once. And then there will also be something called path integration, where you will travel a distance, and when you reach the goal, you will have to shoot a flare or point in the direction you came from. And here you will shoot a flare from where you came from, and you will also be given 1 to 3 stars on this level or on this task. And then lastly, they would also test your reaction, where you will see a sea creature pops up, and then you have to take a picture of it as fast as possible. It also go through a course first, where you have to move from obstacles. So if you want to partake in this study, or might someone you know wants to partake in it, we want people who have experienced CGA and also are comfortable using a phone or tablet, smartphone or tablet to complete this task, and also have access to a smartphone or tablet. And also they need to be able to complete the task in Norwegian or English. And this study is approved by the ethics committee, and there will be no identifiable data stored. Personal data will be kept securely, and participation is entirely voluntary. You can partake in some parts of the study and also not choose to do other parts if that's what you prefer. And you can withdraw with no problem as long as it's before any papers are published with the data. But everything will be anonymized as well. So if you want to partake in the study or know someone who might, you could join easily by, once again, scanning the QR code. And if you haven't experienced TJ yourself, but know someone who might have, you could share the link, QR code, tell them about the study, or just share this event once it is recorded and everything. But now we will move on to a 10-minute break before we will have some people that will come up on the stage and talk about their experience with TJ. And it will also have some questions we can ask them. But yeah, we'll first hear them talk, and then we can ask questions if we have any. But now you can all take a 10-minute break. Now that we're back from the break, we will now hear a little bit about you two sharing your experiences. First of all, we will start with you, Arild, and we will first hear you talk, and then maybe we will have also some questions, and also from the audience, and also if people from the live stream would like to also have some questions, we will also allow for that. So first of all, Arild, you can just start by sharing your experience and telling us a little bit about yourself.
Yeah, I've told something about my medical history. I've written a manuscript, so I'm the only one here with the manuscript to do it, so I think. My name is Arild Bleksund. I'm 65 years old, and I have been married to Sissel for almost 40 years. I work as a professor in quantitative research methods at the Department of Sociology and Social Science, political science here at NTNU. And after a very intense period of work, in the first half of December, I received a TGA on Monday, December 16, 1 week before Christmas. Unfortunately, I have very few memories from that Monday. I know that I was at work at NTNU, campus Dragvoll. During my lunch break, I drove down to IKEA store at Leangen, where I bought 3 lamps and a candle. I found it in my IKEA app, and it was paid for it at 1:13 p.m. After returning to the office, I can now see that I have sent 3 emails to one of my PhD students, one at 2 o'clock, one 2:15, and one at 4 o'clock. That's more a long, extensive email, the last one. I also can see that my wife called me at 1:44, and that the PhD student in question here called me at 2:27. Apart from this, I can't find any trace that I have worked, what I have worked with during the day, and I have probably spent most of the day grading exam answers. I think so, but I have no clear memories. The first thing I remember clearly from that day was when I was driving in the late night for an MRI scan, about 10 o'clock, I think, or something. After that, I can recount everything that happened in detail. So then I returned to a normal state. That means that I have only vague ideas of, or memories from what happened before 4 o'clock that day. And no memories of what happened between 4 o'clock and the MRI scan in the afternoon, perhaps night. And no after effects of TGA after that. So in other words, the problem is I have no memory of when I'm driving home in my car. I know that I came home at around 6 o'clock, and I stopped by the mailbox and picked up the mail. I parked my car in the garage, and I took the shopping bag from IKEA into the living room. And then, when I came in there, my wife, Sissel, told me that I felt weird in my head. I had some problems there. And when we eating dinner, I start repeatedly asking, "Who have been at IKEA?" When I saw the bag from IKEA. And that was a loop, what they call it in the first presentation. Again and again, who have been there? That was new. So she wanted to call the emergency room immediately, but I said that I can take a rest. I can rest on the sofa. I think I will be better then. But after a short time, my daughter, Birgitte, called me from London. She's living there. And she told me afterward that she was upset when I didn't remember that she was married with an Englishman, that she lived in London, and that my 30 years old son, I thought he was still in high school. So she called immediately my wife, her mother, and said, we have to do something here. So they called the emergency room without informing me. And then Sissel helped me put on my clothes. I couldn't do that for myself. And during the trip to the emergency room, I keep asking repeatedly, where are we going? So I had no understanding from about 4 o'clock to 10, perhaps, what was happening around me. It was blocked, everything was blocked. But I also no clear memories from the earlier part of the day. Then you send me some questions, but perhaps you can take them later, or you want to take them now?
I think we're gonna take it like a little bit on the way. Thanks. First and foremost, thank you, Ariel, so much for sharing your story, and what a lucky woman, Sissel, that you can like make such a plan of with time points on exact point, I need to say. But you said that you were talking with her on the phone. Did she say that you sounded?
No, I don't think so. It was a practical thing. I think I can't remember what it was. I think it was something with the data or something. But no, it was nothing. Then she saw it first when I came home at Sitterberg. And but I can't remember that she would call me. That's the problem for me. I'm not sure. And I found the log from the telephone, and I found the logs from emails. That's all I have of information. I'm not clear memories at all.
Yeah. Could you like track back to like any specific event could like start all the series, or do you think it was just like a culmination of all the stress at work and other things around?
I said I had a period with a lot of work before I went. And I think I worked every day the last 3 weeks before the episode. I'm also leader of a local history club. And the 3 Saturdays, we was on a stand and sold annual publication from the history club. And the 3rd Saturday, I had 350 students up to the exam, written exam. So I spent all the time, all the day there. And I think I used the Sunday before I start to graduate. I took the grade exam answers. And the 2 Sundays before there, I prepared a course in a statistical program called R for a research foundation in Trondheim called Ruralis, and should have a course there on Monday, Tuesday, and Wednesday before. So I think
that's
probably that's one
schedule.
Yeah. So it was a hard period with no breaks.
You didn't like localize the last drop, which overfilled the glass in a way, but like it was just like collecting.
That's a problem when you work as a professor, because you have education, or you have lectures before December. In December, you shall do everything that you have not done earlier.
Did all the students pass the exam? Do you remember?
Most of them, I think. But I said that one of my colleagues took that. I had a list of some temporary grades, but one of my colleagues took the responsibility because you gave me 2 weeks off until January. Thank you very much.
I hope you used it well. have you experienced anything anyway similar before? Or?
Yeah, I had one. I can remember it. No, it's not because when I was 10, 12 years old, I fell down from a roof. And then I had a similar repeated questions about what happened now, and I have no personal memories from that situation. So that's it, but it's more than 50 years ago.
It also sounds like amnesia, but maybe a different type of amnesia, I think.
Yeah, I don't know what it is, but that is, I think the symptoms were very simple.
Did you notice any long-lasting effects afterwards? Like, no.
No.
You came back to the baseline immediately.
Then? Yeah. This was very simple. For me, it was a very similar experience.
Great. Livia, do you have...
Did you, like, I know that you've been in Trondheim and everything, but have you had any, like, other follow-up medical assessments post-December?
I've participated in the project, so I invited. Yes, I had 2 memory tests and also scanning. But I don't know. Perhaps you know some results from a scanning. I don't know. I haven't heard anything after that. But no, it's not. I'm no medicals and nothing like that.
Okay, but yeah, thank you so much. If you or if anyone in the audience now will ask questions directly to Arild or...
There we go. Yeah, you mentioned that you wrote 3 emails. Did the emails actually make sense? Were they coherent sentences? Did the emails actually make sense?
Yeah, that was a good question. Because I think I had a conflict with the PhD student in the first. That's the reason why I sent the first email. Because we should have a meeting, and he said that was too windy today, so it's not at campus. So, and then I sent him something, but I have the last email I sent him at 4 o'clock. I gave him some references. I found some actual courses you can take in statistics, and I was a little bit impressed of myself. But I have no memories. But yeah, okay, I have written that.
Okay.
Thank you for your story of the events. I was wondering, you mentioned that you had problems dressing yourself before going out, that you needed help from your wife. Was that correct? And I was wondering, was that because you couldn't remember where your clothes were, or did you have problems remembering how to dress yourself, how to button, how to zip?
I couldn't explain or see the situation around me. So I think it's that's. So I think yes, I parked the car, opened the mailbox, I do everything that. But when she should bring me to the emergency room, I think I was not there at all. So she had to. I couldn't understand the situation at all.
You maybe didn't know why you were going to emergency room, why suddenly they are dragging you somewhere.
I asked all the way from Melhus to Trondheim that, where are we going? And repeatedly. So I was not in the situation.
Thank you so much, Arild. Margit.
Margaret. Hello, Margaret. I'm 56 years old, living here in Trondheim. My experience, last one, I'm the lucky. I'm the special few who has twice experienced this TGA. And the last one was just before 2 or 3 days after you. It was actually, both have started on the night morning. And the last one then, the thing is that on the Wednesday before, I fetched my daughter here. She came home for Christmas to live with us, and we were making lefse on the Wednesday. So, and that's important because I think I remember that there's something strange when I woke up in the morning. And we woke up, me and my partner here, woke up a little bit earlier than usually, at about 6 o'clock. We usually start at 6:45. And it's a kind of early, so we started to do things that partners do when they wake up very early. That might be a trigger, as you say. We don't know. It seems like I think I was in a state of going down into the TJ. I think I remember it because there's something strange going on, but I couldn't fit it. And then afterwards, I went to the bathroom to wash and make my day, and then my partner went to the kitchen to make breakfast, and I used a really long time on the bathroom. Too much time, I really, and then I came to you had to help me with them, but I came to the kitchen. He fetched me and what I'm doing, and I came and then we come to this left side. I was on the kitchen. And I asked, "What is this? When was this made? Who made this? What is this? Who made this?" So I started to repeat questions. And also what is happening and what are you doing, and started to feel that something strange is going on here. As I said, repeatedly asking questions in the loop, as you say, and we found, because I had this TJ before, he knew that okay, this might be the same. And I also maybe mentioned that this might be something like similar to TJ. So we waited till 8 o'clock, called the doctor, went to the doctor, went there, and that's in the morning at around 9 o'clock, 9:10. That's the part where I do remember the least, and came back afterwards, came to the hospital, came to the acute, and I was. Yeah, on the overwalking over when they watched me, and then you came and fetched me. So it wasn't. I didn't feel any like stress, or I didn't feel that it was. I wasn't afraid or anything, and it was the strange thing is that part. I should tell you about the first part afterwards, but this. It was like, it's not completely amnesia. It's more like nearly completely amnesia, because I felt that my brain was trying to remember. It was trying to put things in place. And that's why I think we're just repeatedly asking questions, the same and the same. My brain wanted to remember, wanted to put things in the right positions on what I'm doing. So I do remember more of this last episode than the first one, and also I think I remember more and more afterwards. So it might be that we have talked about it, but I also think that it wasn't completely amnesia, that one. The first one was more deeper. It's happened it's right before the COVID started. This was Saturday, 7th of March. I remember it because it was right before 12th of March in 2020. So I was in Sweden with some friends of mine. We were skiing, downhill skiing, and we went on the Friday. We were out there skiing. I was a little bit tired, and on the morning of Saturday, I woke up, or actually, I do remember that I was standing in the living room with just a blanket around me, and repeatedly asking, what am I doing here? Where am I? What am I doing here? When am I? And then my friend, she understood after a while. Firstly, she thought I was kidding. And then she understood that there's something more going on there. So I remember bits and pieces that she fetched a taxi, got me to the acute there in Åre, and waited there for a long time before it opened. And finally, the helicoptered me to Östersund, where I was in one night, I guess. I was sent home at Monday, so it was the 2 nights. And that that I found really interesting, because when I was waking, when I coming to myself, it was like I remembered memories, deep down memories that I haven't been remembered. It wasn't action, it wasn't the actual thing, it was the memory I was remembering, I was feeling again. And it was like deep down, and I realized that this one is this memory I haven't been thinking about for many years, like in the teenage years. And then suddenly there's something above, there's some other memory laying itself above that. And then I remembered something a little later on, forgetting that first memory. And then it came a new memory laying above that again. It was like memory stacking itself on top of each other. So I forgetting those old memories. That was really interesting. So I find these experiences really interesting, fascinating. I love it. I wasn't afraid. I think this is so fascinating to have experienced this. So. And I was the first. Then it was, as I said, it was a little bit stressful that day before, but the last one in December, I wasn't that stressed. I have been a quite stressful period, October, September, all the year, but after that it has been quieted down. So I didn't remember that it was so much a stressful period right before. So I don't know why it happened. But both started in the nighttime, morning.
So for the 2 different episodes, did you experience any other warning signs before for any of the 2 episodes, or? No. I haven't. So it just happened. So, yeah. And you remember or you said that the first one was kind of deeper than the second one.
Felt deeper. Yeah. Felt more like global than this last one was. Who wasn't? The last one wasn't that global. It was more. I remember more during the episode, all of it, because I remember more from where put in the waiting room at the doctor's. And then I forget something I don't remember, and then I remember some. But it's really that also the feeling that my brain wanted to remember. It's really trying hard to remember, to put the things around me into system, into where it should be, and wasn't able to do that. It was like a control room. Doesn't work, and trying to make it work, and started. Oh no, it's working. Oh no, it's not working. No, it's working. Oh no, it's so really. Yeah. that's why I think it's really interesting. It wasn't dangerous. I didn't felt any. I wasn't afraid at any time.
But yeah, for both of the episodes, did you like experience any lasting effects like after it? Or
No. My memory is decreasing, but that's because I'm older than I was before. And also in my menopause, and that's also so that's. But I don't think it's any TGA experience.
That part you're saying that you could like access like a deeper memories you didn't have access to. It's for a person being a researcher of memory, it's very fascinating to hear. Is there anything unusual which happened just afterwards, or you could recall? No.
No, I was just laying there in the bed, in the hospital bed in Östersund, and the joy. It's actually the joy of remember those old memories, and the grief of losing them again. That was okay. But it was really a strange and interesting experience, that how it disappeared down, and something else came up, and then it disappeared, and then a new thing disappeared, and then I came back to.
May I ask a question about
yes,
that?
of course.
So that stacking of the memories, were those older memories stacked below the more recent ones? So it was really a timeline through your life, or fascinating, yeah.
So I can't tell you now what they were because they have disappeared. So it was like it was as on, this memory I haven't been thinking about for a long time, and then it disappeared, and something else came up, and it wasn't that many things, but it was like I think it was like some important memories about something I've been. Thinking about felt some joy with, and then so just like my brain wanted to remember those. I can't explain it more like that than that.
Because you kind of remember that they were joyful. That you remember the emotions with the memories.
Emotions. I remember the emotions. Yes.
And Adel, you were nodding. Did you also have some related experience, or?
Yeah, I haven't thought about it, but I think I had more, I remember more of the situation before 4 o'clock, the day after, I think, than I do now. Wow. But that's because you said to me that I will reconstruct, the memory will come back until before o'clock, but after 4 o'clock to up to 10 o'clock, nothing. I have no memories, I've never been there. But the strange thing is that the earliest day, from the morning until 4 o'clock, I think I had no normal situation, I think, but it's impossible for me. And I have some small fragments I can remember, but I'm unsure, is it true or is it something I constructed afterwards? So I don't know.
You were talking about how you've had one in Östersund and then you had the one during December. So you had 2, and you mentioned that the second one didn't feel as deep as the first one. So more towards maybe the clinicians, if you have any patients who've had 2 TGAs, if they have similar experiences with, are they less intense the second time around or not?
Right out of the head, I cannot recall, but yeah, maybe there's anybody who had TGA in here who can say something. Did they?
I had twice. The first one was very deep, as you say, and I've lost about 3 days they never came back. Second time, I think I gained most of the lost memories, and I think I remember everything now. But the first time it was very scary because I lost so much. 3 days. I could remember which day it was, but I couldn't remember the date, and that was about 12 hours later. So it took some time to get my balance, and I kept asking for many hours, and that was kind of scary for my family because we thought I had a stroke, or maybe when I got to the hospital, maybe I was something else, Alzheimer or whatever, but they assured me it was not. And this amnesia was something the doctor didn't know about. He had to Google it to find out what this was. Yeah, and this was 2016, I think. So, and both my attacks was at the same year. So, and as you say, the first one is very deep. Next one, kind of waking from a dream. Yeah.
Thank you. Thank you so much.
I have a question, but not about the past. I'm a little curious about now and the future. For example, after that event, what happened in your life? Are you started to recalling your memory more often than before? Or is there any things that you're feeling that if this happens, maybe I will feel the same, or any triggers? Are you doing lots of the flashback to the before? For example, again, remembering all the memories from the back, or just living your normal life, something has never happened to you?
Are you asking me?
In general.
You can answer first, I think.
I'm not sure if I can, but the thing that are gone is gone. I don't remember those. And those things that those memories I told you about, they are gone. Those older memories that are gone. So, and I'm living my normal life. I don't think that I'm going to experience this again. I don't know. But if I do, I won't get afraid, I guess. So, but it was that my friend, she was so tired afterwards, repeatedly answering my questions, the same, because they were in a loop. So she said that afterwards that she found she didn't talk badly to me, but she what do you call it? She
She got slightly pissed.
Yes. And then she said, I can say this to you because I know you won't remember. And she told me that afterwards. So, but they were scared. It was I wasn't scared. I remember I was put in a helicopter to go to Östersund because they thought I was having a stroke, or they should, one of the theories. So I felt really, so I was happy flying with a helicopter, taken care of. I didn't know what's happening, but I was calm, Mrs. So yeah.
Do you remember any of the helicopter flights?
Yes,
right
of
now?
course. I was watching the nature, was snow, and it was so nice. So fun.
Perhaps we could take a question online, because there are actually a lot of people that are asking questions also here on the chat. And maybe a question for you, Livia. Have you found, and Alexandra, of course, have you found that some people struggle cognitively after the TGA? And if so, does it resolve itself after time? After 9 months, I still feel slower, especially during conversation. What do you think about this? And is there something we can do to help with that, perhaps, to find out?
This is, of course, one of the things that we want to investigate with our online study, because we want have patients who just recently experienced TGA. We will have people who experienced it even years, months ago. So we will then look into if there are any, if they feel that there are any, and also we'll be able to in some way assess whether there are any cognitive impairments after the episode as well. But you have more clinical experience, so
yeah, from my observations, there is quite a big group of people who say, I cannot put a finger on it, but I'm thinking a little bit more slowly. My memory isn't exactly the same. That something is still wrong. And I meet most of those people 14 days after, but I was also called by some of the patients that something is still wrong. So that they don't feel exactly the same like they did before the episode. So yeah, absolutely.
I can say something too, because I think I have no after effects, I think. And I remember the day after my TGA, I took a memory test. And my impression was that, oh, they will be so impressed. But I haven't seen the scores.
We keep them the same. But in the cognitive tests we are performing on the participants, we can see with naked eye that there is a difference between 24 hours later, where it's supposedly be totally gone, till 14 days later. So from my experience at least, there is more to TGA than just those 24 hours.
And there was also a question over there.
I had a TGA for exactly 3 years ago, and Easter. and I wonder if you feel the same as I do after I went home from the hospital, because I want to remember everything I say, everything I hear, everything I saw. And I work in a kindergarten in Melhus. And I start to, who's the kid? Everyone kid, I want to remember all. And I do that like this for 2 months. It was very stressful, because I want to convince myself that I don't lost my memory. Do one of you feel like that?
Have you seen the results of my 2 memories? No, I had nothing after December 17.
Yeah, I don't think that I had the necessity to remember more afterwards. I don't think, but maybe I feel a little bit that when I talk, and it might be after the last one. I don't remember. I don't know when things started, because, as I said, I'm experiencing to be in the menopause, and there's a lot of things happening there. Also with I feel thinking a bit slower. I sleep not that well as before, so that's affecting me. But I feel that when I discuss things, when I'm talking about things, then I sometimes have to stop to wait to have my words coming back after me. That what is that? How to explain this in English? In the discussions, it's like my mouth has to wait until my brain is coming to my mouth. So the words are coming to my mouth. So I need to stop more often to finish the sentences, to continue the sentences. That might be some TGA effect. I don't know, or it might be that I'm in my age. But I'm not. I didn't. It was right before Christmas. It's like the house was filled up, and there was a lot of food and dinner making and people, and so I don't think I had the time to think about that, to how to remember more.
That's also my situation, because I was off job the next 2 weeks, and it was Christmas. The total life was totally different than before. So
it's very interesting what you are all talking about. It's almost like you desperately try to grab to your own memories to not lose them again.
Yes, because the 24 hours I was gone is gone forever, I think. So it was very deep, these 24 hours. But afterward, I tried to remember everything, and that was very stressful for 2 months. And then it's go off and off and off.
May I ask a question? Does it work? Microphone?
Yeah.
Yeah. Okay. You were talking about TGA and Alzheimer as kind of similar experiences. So I was wondering whether of the patients who had had TGA, do you find that there is an increased likelihood of developing Alzheimer?
I guess I can just answer, and maybe we will wrap up because we are arriving to 9. There has been some studies, and these studies are epidemiological studies with a lot of, statistics to understand correlation. But correlation does not say much about causation. So things point that there is perhaps some kind of correlation to develop either epilepsy or cognitive difficulties, but it's kind of speculative at this point. And that's also a question that came back online. So I want to reassure everyone, we don't want to establish a link here. However, I think with the tests that we've built up online, we'll be able to maybe capture subtle cognitive changes, which I think will be meaningful to understand a little bit more about this process. First, to help patients, and second, to understand how we can use TGA as a window towards memory. And when we do so, then we can also study memory, chronic memory deficits, perhaps like Alzheimer's and others. And we are quite well placed to do this here. So on these words, I think we're arriving to 9 p.m. I want to thank everyone for your time and attention. I want to give a big round of applause also to the speakers that came here, and also encourage again to all the speakers, the participants online that will watch also the stream later, to scan the QR code and perform this cognitive testing. So that's the best way you can help us to understand TGA. Thank you.